Continuando con el programa de hoy, a nombre del señor embajador de la UNESCO, Jorge Herrera Ramos, y chairman del World Congress of Neurosurgery, UNESCO ASPNet, Walter E. Dandy, in honor of Professor Yaşargil, a quien agradecemos por la organización de este magno evento, así como a la doctora Deyanira Capi Casillas, chairman del capítulo ANSI-PN, y al doctor Salim Abdulrauf, chairman de Walter E. Dandy Neurosurgical Society, y al Dr. Babak Atef, chairman de la Society for Brain Mapping and Therapeutics y de Brain Mapping Foundation, todos ellos vice-chairmans del comité organizador, y al Dr. Parménides Guadarrama Ortiz, chairman del Centro Especializado de Neurocirugía y Neurociencias México. Es para mí un honor presentar la siguiente ponencia a cargo del Dr. Michael Bassil, quien es estudiante de la Universidad de Medicina y Química Farmacéutica Carol Dávila de Bucarest, Rumania, con el tema de esclerosis lateral amiotrófica. Dr. Sebastián.
Good afternoon or morning, depending on where you are listening from. I'm Dr. Sebastian Dobrescu, and it is my pleasure to open today's presentation. Before we start, I want to welcome you to the third week of the World Congress of Neurosurgery, UNESCO, Walter E. Dandy Society, in honor of Professor Gazi, a surgeon. I don't hear anyone. On behalf of the Ambassador of UNESCO, ASPNet, Jorge Herrera Ramos, President of the World Congress, whom we thank for organizing this event, as well as the Vice Presidents, Dr. Deyanira Capi, Presidents of the Untitled Chapter, Dr. Salim Abdulrauf, President of Walter E. Dandy Neurosurgical Society, Dr. Babak Atef, President of the Society for Brain Mapping Therapeutics, Brain Mapping Foundation, Honorary Chairman of the Committee, Professor Nasser Al-Gandour, Professor Marcus Rota, and to Dr. Parmenides Guadarrama Ortiz, Director of the Specialized Center for Neurosurgery and Neurosciences of Mexico. It is an honor to present today's mediator, Dr. Cristina Sari.
Thank you, Dr. Sebastian Dobrescu. It is a pleasure for me to be here today again with you, and it's my honor to present today's presenter, Dr. Michael Bassil. He's a member of the Romanian Association of Neurosciences and a student at Carol Dávila University of Medicine and Pharmacy of Bucharest, Romania. With the topic today, amyotrophic lateral sclerosis, welcome, Dr. Michael Bassil.
Thank you for welcoming me. I will now be presenting amyotrophic lateral sclerosis. Can you see?
Yes, we can see Dr. Bassil.
Thank you. So amyotrophic lateral sclerosis, also known as ALS. ALS belongs to a wider group. ALS belongs to a wider group of disorders known as motor neuron diseases, which are caused by gradual deterioration or degeneration and death of motor neurons. Motor neurons are nerve cells that extend from the brain to the spinal cord and to the muscles throughout the body. These motor neurons initiate and provide vital communication links between the brain and the voluntary muscles. Messages from motor neurons in the brain, called upper motor neurons, are transmitted to motor neurons in the spinal cord and to motor nuclei of the brain, called lower motor neurons, and from the spinal cord and motor nuclei of the brain to a particular muscle or muscles. The upper systems present spasticity, exaggerated reflexes, and lower systems, wasting, fasciculations. There are no sensory symptoms present in ALS. Some cranial nerves can be involved, as seen in dysphagia. Dysphagia is trouble swallowing. In ALS, both the upper motor neurons and the lower motor neurons degenerate or die and stop sending messages to the muscles. Unable to function, the muscles gradually weaken, start to twitch, called fasciculations, and waste away, called atrophy. Eventually, the brain loses the ability to initiate and control voluntary movements. Most people die from respiratory failure within 3 to 5 years. Glutamate. So nerve cells pass signals to each other and to their target organs by releasing messenger molecules called transmitters. Many are simple amino acids, such as the one called glutamate. The message is intended to tell the recipient neuron whether to fire off its own neurotransmitters. As with all neurotransmitters, glutamate docks at specific recognition molecules on the receiving neuron. Glutamate is then cleared from the nerve cell junctions to keep the message brief. Prolonged excitation is toxic to nerve cells, and neurobiologists recognize that glutamate can cause harm when the messages are overwhelming, as in stroke or epilepsy. Glutamase toxicity is due to calcium flooding the cell. Calcium is supposed to briefly enter the neuron with each signal and triggers the cell to fire off its own signals and adjust its own activities accordingly. But prolonged calcium inside the cell evidently can do damage and will even activate programmed cell death. The transporter that clears glutamate is called EAAT2, an excitatory amino acid transporter, as glutamate is one of the amino acids that serve as transmitters. In ALS, transport of glutamate is slowed into the glial cells that surround the junctions of motor neurons. Indeed, a mutation has been identified in an ALS patient that prevented the transporter from working properly. Excitotoxicity is propagated primarily through the calcium permeable receptors. An influx of calcium is buffered by the endoplasmic reticulum and the mitochondria, but in the presence of excess calcium influx, these systems can be overwhelmed. A calcium overload or perturbations of intracellular calcium compartmentalization can activate or enhance mechanisms leading to cell death. An imbalance between calcium influx and efflux from cells is the initial signal leading to calcium overload and death of neurons. In addition, alterations in intracellular calcium storage can integrate with death signals that do not initially require calcium to promote processing of cellular components and death by apoptosis or necrosis. Finally, calcium can directly activate catabolic enzymes such as proteases, phospholipases, and nucleases that directly cause cell demise and tissue damage. When the mitochondrial buffering system fails, the cell becomes highly vulnerable to mitochondria-mediated apoptosis, reactive oxygen species, and production and electron chain dysfunction. So in the US, ALS is mostly known as Lou Gehrig's disease. Lou Gehrig was a baseball player that played for the New York Yankees. And in the medical world, ALS became more known through him being diagnosed with it. At first, they did not know what he had. And he died 3 years after being diagnosed with ALS. The Ice Bucket Challenge, sometimes called the ALS Ice Bucket Challenge, was an activity involving the pouring of a bucket of ice water over a person's head, either by another person or self-administered, to promote awareness of the disease. The challenge encourages nominated participants to be filmed having a bucket of ice water poured on their heads, and then nominating others to do the same. A common stipulation is that nominated participants have 24 hours to comply or forfeit by way of charitable financial donation. On July 25th, 2016, the ALS Association announced that, thanks in part to donations from the Ice Bucket Challenge, the University of Massachusetts Medical School has identified a third gene that is a cause for the disease. Project MIN-E, a global gene sequencing effort to identify genetic drivers of ALS, received $1 million from the challenge, allowing them to broaden the scope of their research to include new sources in new parts of the world. Having identified the link between the gene NEK1 and ALS will allow for a new targeted gene for therapy development as well as focused drug development. The challenge raised over $220 million worldwide and over 2.4 million tagged videos on the internet. So who can get ALS? There are mainly two types of ALS, one being sporadic ALS. The majority of ALS cases, about 90%, are considered sporadic. This means the disease seems to occur at random with no clearly associated risk factors and no family history of disease. Although family members of people with sporadic ALS are at an increased risk for the disease, the overall risk is very low and most will not develop ALS. In familial ALS, it accounts for about 5 to 10% of all ALS cases, which means that an individual inherits the disease from his or her parents. The familial form of ALS usually only requires one parent to carry the gene responsible for the disease. Mutations in more than a dozen genes have been found to cause familial ALS. About 25 to 40% of all familial cases are caused by a defect in a gene known as chromosome 9 open reading frame 72, or C9orf72. Interestingly, the same mutation can be associated with atrophy of frontal temporal lobes of the brain, causing frontal temporal lobe dementia. Some individuals carrying this mutation may show signs of both motor neuron and dementia symptoms. Another 12 to 20% of familial cases result from mutations in the gene that provides instructions for the production of the enzyme copper zinc superoxidismutase 1, or SOD1, which I'll talk about later. It is also seen to be higher in military veterans. We'll discuss this later as well. So symptoms. For many individuals, the first sign of ALS may appear in the hand or arm as they experience difficulty with simple tasks, such as buttoning a shirt, writing, or turning a key in a lock. In other cases, symptoms initially affect one of the legs, and people experience awkwardness when walking or running, or they notice that they are tripping or stumbling more often than normal. When symptoms begin in the arms or legs, it is referred to as limb-onset ALS. Other individuals first notice speech or swallowing problems termed bulbar-onset ALS. Regardless of where the symptoms first appear, muscle weakness and atrophy spread to other parts of the body as the disease progresses. Individuals may develop problems with moving, swallowing, also known as dysphagia, speaking or forming words, dysarthria, and breathing, dyspnea. Although the sequence of emerging symptoms and the rate of disease progression vary from one person to another, eventually individuals will not be able to stand or walk, get in or out of bed on their own, or use their hands and arms. Individuals with ALS usually have difficulty swallowing and chewing food, which makes it hard to eat normally and increases the risk of choking. They also burn calories at a much faster rate than people without ALS. Due to these factors, people with ALS tend to lose weight rapidly and can become malnourished. Because people with ALS usually retain their ability to perform higher mental processes such as reasoning, remembering, understanding, and problem solving, they are aware of their progressive loss of function and may become anxious and even depressed. A small percentage of individuals may experience problems with language or decision making, and there is growing evidence that some may even develop a form of dementia over time. Individuals with ALS will have difficulty breathing as the muscles of the respiratory system weaken. They eventually lose the ability to breathe on their own and must depend on a ventilator. Affected individuals also face an increased risk of pneumonia during later stages of disease. Besides muscle cramps that may cause discomfort, some individuals with ALS may develop painful neuropathy, a nerve disease or damage. No one test can provide a definitive diagnosis of ALS. ALS is primarily diagnosed based on a detailed history of the symptoms and signs observed by a physician during the physical examination, along with a series of tests to rule out other mimicking diseases. However, the presence of upper and lower motor neuron symptoms strongly suggests the presence of the disease. Physicians will review an individual's full medical history and conduct a neurologic examination at regular intervals to assess whether symptoms such as muscle weakness, atrophy of muscles, and spasticity are getting progressively worse. ALS symptoms in the early stages of the disease can be similar to those of a wide variety of other more treatable diseases or disorders. Appropriate tests can exclude the possibility of other conditions. Muscle and imaging tests, an electromyography, an EMG, a special recording technique that detects electrical activity of muscle fibers, can help diagnose ALS. Another common test is a nerve conduction study, NCS, which measures electrical activity of the nerves and muscles by assessing the nerve's ability to send a signal along the nerve or to the muscle. Specific abnormalities in the NCS and EMG may suggest, for example, that the individual has a form of peripheral neuropathy, which is damage to peripheral nerves outside of the brain and spinal cord, or a myopathy, muscle disease, rather than ALS. A physician may also order a magnetic resonance imaging, MRI test, a non-invasive procedure that uses a magnetic field and radio waves to produce detailed images of the brain and spinal cord. Standard MRI scans are generally normal in people with ALS. However, they can reveal other problems that may be causing the symptoms, such as spinal cord tumor, a herniated disc in the neck that compresses the spinal cord, a syringomyelia, a cyst in the spinal cord, or cervical spondylosis, abnormal wear affecting the spine and the neck. There are also laboratory tests. Based on the person's symptoms, test results, and findings from the examination, a physician may order tests on blood and urine samples to eliminate the possibility of any other diseases. There's also tests for other diseases and disorders, infectious diseases such as human immunodeficiency virus, HIV, human T-cell leukemia virus, polio, and West Nile virus can, in some cases, cause ALS-like symptoms. Neurological disorders such as multiple sclerosis, post-polio syndrome, multifocal motor neuropathy, and spinal and bulbar muscular atrophy also can mimic certain features of the disease and should be considered by physicians attempting to make a diagnosis. Fasciculations and muscle cramps also occur in benign conditions. Because of the prognosis carried by this diagnosis and the variety of diseases or disorders that can resemble ALS in the early stages of the disease, individuals may wish to obtain a second neurological opinion. From a microscopic point of view, microscopic changes include neuronal and axon loss. There is a loss of myelinated axons in the lateral and anterior columns of the spinal cord and decreases in size of anterior horn of the spinal cord, best shown by myelin stains such as Luxol-Fast blue. There is degeneration and loss of the large motor neurons in the anterior horn of the spinal cord, lower cranial motor nuclei of the brainstem, and bed cells in the motor cortex, best seen with routine stains such as hematoxylin and eosine. Morphometric studies of the spinal anterior horn have shown a global reduction of all neurons in the anterior horn, not just the large alpha motor neurons. There is evidence of reduction in neuron size as well as loss and atrophy of nerve fibers. Other pathological features of ALS include vacuolization, large empty spaces near neurons, and spongiosis, microscopic holes resulting in a sponge-like appearance. And here, A and C are with ALS, and B and D would be the control without ALS. Lateral sclerosis is shown in thoracic spinal cord and sporadic ALS in A, and compared to control B. The inserts show loss of myelin in the white matter tracts. Loss of motor neurons is shown in the lumbar spinal cord of sporadic ALS in C, and compared to control D. The inserts show the motor neurons in the anterior horns under higher power, using Luxol-Fast blue with hematoxylin and eosine for A and B, and hematoxylin and eosine for C and D. Covalent binding of ubiquitin to proteins marks them for degradation by the ubiquitin ATP-dependent pathway, right here. This pathway plays a major role in the breakdown of abnormal proteins that result from oxidative stress, neurotoxicity, and mutations. Failure to eliminate ubiquitinated proteins disrupts cellular homeostasis, causing degeneration. Inclusions containing ubiquitinated proteins are commonly detected in many neurological disorders. In 1988, when Ley and Loud independently discovered ubiquitin-positive skein-like or dense round structures in the cytoplasm of anterior horn cells in both familial and sporadic ALS, inclusions that are not detected by H&E and other routine staining methods. Such inclusions were later identified in frontotemporal lobar degeneration and became the cornerstone of distinguishing between frontotemporal lobar degeneration with ubiquitinated inclusions from frontotemporal dementia with TAU and other inclusions in both ALS and frontotemporal lumbar dementia. The ubiquitin positive inclusions have been observed in neurons of the frontal cortex, temporal cortex, hippocampus, and striatum. Although they are most commonly found in neurons, they have occasionally been seen in glial cells. They are negative for proteins commonly associated with neurodegenerative inclusions such as TAU and alpha-synuclein. TAR DNA binding protein 43, also known as TDP43, was linked to ALS in 2008. Mutations in TDP43 cause a dominant form of ALS and are responsible for about 4% of familial ALS and about 1% of sporadic ALS. Mutations in the TDP43 gene cause the TDP43 protein to mislocalize in motor neurons, away from the nucleus where it is normally found and into the cytoplasm, the material surrounding the nucleus, where it aggregates into clumps that can be seen under the microscope. The presence of ubiquitin-positive inclusions suggested a problem with some other proteins. 18 years later, TDP43 was identified as the main component of ubiquitinated inclusions in both ALS and FTD patients. This connected ALS and FTLD as TDP43 proteinopathies. TDP43 is a heterogeneous nuclear ribonucleoprotein and has many different cellular functions, including mRNA stability, mRNA processing, mRNA transport and translation, and negative regulation of alternative splicing. Under normal conditions, TDP43 is expressed in many tissues, including the nuclei of neurons and glial cells. In sporadic and most familial ALS, as well as frontotemporal dementia TDP43, there is loss of nuclear TDP43 and formation of pathological aggregates in the cytoplasm. The mechanism behind this redistribution is poorly understood and could be either the translocation of TDP43 from the nuclei to the cytoplasm or an impaired TDP43 cytoplasm to nucleus shuttling process. Indeed, antibodies against TDP43 phosphopeptides stain ubiquitinated TDP43 positive inclusions in frontotemporal dementia and ALS patients. It is commonly believed that motor neuron death in ALS closely resembles apoptosis, although the evidence for this is incomplete. One researcher postulated 3 stages of neuronal death in motor neurons of ALS patients: chromatolysis, which is the dissolution of the initial bodies in the cell body of a neuron, somatodendritic attrition, and finally apoptosis. This process of neuronal death is accompanied by morphologic findings such as cytoplasmic and nuclear condensation and darkness. DNA fragmentation in the presence of DNA fragmentation activation factor, as well as a lack of appreciable vacuolar and edematous cytoplasm in dying neurons. Based on these observations, he concluded that apoptosis plays a role in neuronal death in ALS cases. Staging of ALS neuropathology, similar to Alzheimer's and Parkinson's disease, has been proposed by Brack. In this stage 1 disease is characterized by mild burden of TDP-43 pathology involving motor cortex, brainstem, motor nuclei, and spinal motor neurons. Stage 2 disease involves mild moderate burden of TDP-43 with dissemination into prefrontal neocortex, reticular formation, and precerebral nuclei. Stage 3 disease involves moderate burden of TDP-43 with dissemination into basal ganglia and prefrontal and postcentral neocortex and striatum. Stage 4 disease involves severe burden of TDP-43, including the hippocampal formation. Genes are strings of DNA that provide instructions or code for making proteins. Proteins are the principal components of all types of cells, controlling reactions and providing structure. Genes are linked together to form chromosomes, which reside in the nucleus of each cell. Chromosomes and the genes they contain are inherited, passing from parent to child. A mutation is an error in the DNA instructions, usually causing the cell to make either too little protein, too much protein, or a defective protein. Different mutations may cause different effects. Any change in the normal protein can be harmful to the cell and may cause disease. For instance, loss of function of the angiogenin protein due to mutation in the gene is thought to be linked to some cases of ALS. A gain of a new and toxic function of the superoxidismutase 1, SOD1 protein, is probably how mutation of the SOD1 gene causes ALS. A mutation may also be harmful, not because it changes the protein encoded by the gene, but due to its effect on RNA. One type of RNA, called messenger RNA, is an intermediary or messenger between gene and protein. To make a protein, the cell first uses the DNA gene to form an mRNA copy. That mRNA copy is then used to provide the working instructions to make the protein. After it is formed and before it is used to make protein, RNA is processed in several different ways. Mistakes in RNA processing may cause disease. For instance, the C9orf72 gene may cause ALS due to accumulations of RNA that occur when the gene is mutated. FUS and TDP-43 mutations may impair the normal processing of RNA from a wide variety of genes, leading to ALS. Genes are present in pairs, one copy inherited from each parent. For most ALS genes, only one mutated copy is needed to cause disease. This is called a dominant gene. For other genes, both copies must be mutated to cause disease, and this is called a recessive gene. If a parent carries one copy of the disease gene, there's a 50-50 chance of passing along the disease gene to any one child. If the disease gene is dominant, the child will develop the disease, since only one copy is needed. If the gene is recessive, the child will need to inherit 2 disease genes, one from each parent. If both parents carry one copy of this recessive gene, the chances of any one child inheriting 2 copies and developing the disease is 25%. ALS is remarkably similar whether it is inherited or appears in a person spontaneously with no family history of the disease. Therefore, insights gained from studying genetic forms of the disease are likely to benefit those with sporadic ALS as well. Therapies aimed at correcting the consequences of gene mutations may lead to treatment for all cases of ALS. The ALS Association has made significant investments into identifying new genes for ALS and has supported all the major gene discoveries. The 2 landmark discoveries are the SOD1 gene mutations in 1993 and C9orf72 in 2011, the most common gene associated with ALS. Superoxidismutase, also known as SOD1, is an enzyme comprised of 153 amino acids that in humans is encoded by the SOD1 gene located on chromosome 21. SOD1 is one of 3 human superoxidismutases, an enzyme that alternately catalyzes the dismutation of the superoxide, O2 negative, radical into ordinary molecular oxygen, O2, and hydrogen peroxide, H2O2. Superoxide is produced as a byproduct of oxygen metabolism and, if not regulated, causes many types of cell damage. Mutations in the superoxidismutase 1 gene account for 20% of all familial ALS cases. To date, over 180 different mutations have been described, including single-point mutations, deletions, insertions, and truncation mutations throughout the 5 exons of the SOD1 gene, pointing out that SOD1 toxic effects are the results of protein failures due to the misfolding of the protein. The most common mutations found in the SOD1 gene are the D90A, the A4V, and the G93A. The D90A, the aspartic acid at codon 90 changed to alanine, is the most common mutation and can be inherited as both a dominant and a recessive trait, although in the majority of cases it is recessive. The A4V, alanine at codon 4 changed to valine, is the most common, approximately 50% of ALS causing mutation in the US population. The G93A, glycine 93 changed to alanine, is indeed a relatively rare mutation. Nevertheless, it has been investigated very carefully as it was the first mutation to be studied in a transgenic mouse model where it is able to generate motor neuron syndrome. Several transgenic murine lines for SOD1 have been generated in knockout mice deficient in SOD1 and mice bearing a human transgene of familial ALS associated mutant SOD1. For example, mice carrying the G85R mutation exhibit a reduced lifespan and display SOD1/ubiquitin positive aggregates. It is widely accepted that a hallmark of SOD1 associated ALS is the deposition of SOD1 into insoluble aggregates in motor neurons, probably due to a consequence of structural destabilization and/or oxidative damage induced by gene mutations, which in turn contribute to the misfolding and aggregation of SOD1 into neurotoxic species. The C9orf72, or chromosome 9, open reading frame 72, is a protein which in humans is encoded by its own gene, the gene C9orf72. The protein is found in many regions of the brain, in the cytoplasm of neurons, as well as in presynaptic terminals. Disease-causing mutations in the gene were first discovered by 2 independent research teams led by Rosa Raidmakers of Mayo Clinic and Brian Terrainer of the National Institutes of Health, and were first reported in October 2011. The mutations in C9orf72 are significant because it is the first pathogenic mechanism identified to be a genetic link between familial frontotemporal dementia, FTD, and of amyotrophic lateral sclerosis, ALS. It is the most common mutation identified that is associated with familial FTD and ALS. This not only linked ALS and FTD at the genetic level, but connected them to repeat expansion diseases. The mutation of C9orf72 is a hexanucleotide repeat expansion of the 6-letter strings of nucleotides GGGGCC. In a person without the mutation, there are few repeats of this hexanucleotide, typically less than 20 to 30. But in people with the mutation, the repeat can occur in the order of hundreds. It is known that the mutation interferes with normal expression of the protein made by C9orf72. However, the function of this protein remains speculative. There are 3 major theories about the way that C9orf72 mutation causes FTD and/or ALS. In A at the top, the C9orf72 gene harbors a polymorphic hexanucleotide, 4GCC, repeat in a non-coding region of the gene. Large expansions of this nucleotide repeat cause C9 FTD and ALS. There are currently 3 major hypotheses to explain how such repeat expansions could be pathogenic. In B, the large GGGCC repeat expansion could cause a downregulation in C9orf72 gene expression by interfering with transcription, leading to a decrease in C9orf72 protein and a loss in C9orf72's function. In C, RNA transcripts harboring C9orf72 repeat expansions are produced by both sense and antisense transcription, resulting in the accumulation of nuclear or cytoplasmic foci of 4GCC RNA, as well as the antisense RNA, which could cause a sequestration, the detachment of essential RNA binding proteins, including splicing factors. During splicing, introns that are non-coding regions are removed, and exons that are coding regions are joined together, leading to defects in pre-mRNA splicing by an RNA toxicity mechanism. And indeed, sense and antisense repeat RNAs are substrates for an unconventional form of translation to generate a series of dipeptide repeat proteins, which accumulate in the brain and spinal cord of C9orf72 mutation carriers and may cause disease by dipeptide repeat protein toxicity. In 2009, mutations in the RNA binding protein fused in sarcoma, FUS, in a subset of familial ALS patients were identified. FUS mutations are responsible for 5% of familial ALS cases and are distinct from TDP43 proteinopathies, but they follow a similar motif where a protein involved in RNA metabolism is mislocalized from the nucleus and aggregates in the cytoplasm of neurons. Mutant FUS forms large ubiquitinated TDP43 negative neuronal cytoplasmic inclusions and occasional neuronal intranuclear inclusions in the spinal cords and brains of affected patients. These inclusions take the form of fine and coarse granules as well as filaments and can be seen in neurons and glia. They are thought to interfere with RNA processing and cause the formation of cytoplasmic stress granules. In contrast with TDP43, NCIs, cytoplasmic FUS aggregates and non-pathogenic nuclear FUS are not exclusive and can be observed in the same cell. Discovered in 2016 through a global effort by Project MinE, as discussed earlier, mutations in NEK1 are present in both sporadic and familial forms of ALS. Together, NEK1 is associated with 3% of all ALS cases. Research is currently ongoing to understand the role of NEK1 in ALS disease, how it is inherited, and other important questions that we should be asking ourselves. The NEK1 gene is among other genes whose role in disease is much less understood. It has been proposed that variances, differences in DNA sequences in this gene result in a severe disruption in the NEK1 protein function, increasing the risk of ALS significantly. NEK1 works to control cell division and repair DNA errors and regulates the formation of cilia, projections from cells that sense the environment, and the function of mitochondria, the cell's powerhouse. Currently, researchers do not know how the NEK1 gene is inherited, its penetrance, an example, the proportion of individuals with the NEK1 mutation that show ALS symptoms, or whether the mutation is sufficient alone to cause the disease. Researchers are now working diligently to answer these significant questions. Biomarkers are by definition any measurable substance that changes in quantity or that appears or disappears with a change in the body's state. Examples of these types of biomarkers are chemical changes in the blood, the urine, or cerebral spinal fluid that bathes the brain and spinal cord. In addition, structures in the body that change with disease's state could serve as biomarkers. Biomarkers that serve as a diagnostic tool should be specific to ALS, whereas biomarkers that inform about disease progression should be sensitive enough to demonstrate changes during the disease process and are valuable tools to assess whether a drug is impacting the disease process of interest. Methods now available make it possible to analyze very small amounts of fluid collected from living beings. Charged surfaces of protein-binding chips can separate all the different proteins found in the blood, CSF, urine, or other body secretions. Investigators can use an extremely sensitive technique to identify the separated molecules called mass spectrometry. Other routes to ALS biomarker discovery would be imaging studies that can show that certain changes in the brain or spinal cord accompany the disease and are specific to ALS. Imaging methods such as magnetic resonance imaging, MRIs, and positron emission tomography, PET scans, promise the specificity and sensitivity that are required to produce an ALS diagnostic marker. Measures of muscle strength and force vital capacity are traditional measures that are used in clinical trials. Together, any measure that changes with ALS and or is specific for ALS and will not confuse ALS with another disorder could serve as a biomarker for the disease. Biomarkers have multiple essential functions. For example, they can be used to follow disease progression and track response to therapy. They are important to understand if a drug is hitting its target and is a marker of whether or not drug is working. Biomarkers could also be used to accurately detect ALS earlier and speed up diagnosis, allowing a greater chance for people to seek earlier treatment to alter disease course. Currently, it takes an average of 1 year to diagnose a person with ALS and relies on ruling out ALS mimics and confirming the disease as it progresses. This delay in diagnosis hampers drug discovery and the development of new treatments, making it more difficult to rescue the cells that are dying in this disease. There are many clinical variants of ALS, so a form or version of something that differs in some respect from other forms of the same thing or from a standard, that appear to be distinctive, and a key debate is whether these are distinct disease entities with different biologies or ends of a continuum. The neuropathological evidence is minimal, but suggests that they share a similar neuropathology, and differences are based on the anatomical distribution of the pathological burden rather than biological differences. Separate from this debate about ALS are the important genetic syndromes that affect the motor system but are clearly different from ALS. Since these may be confusing, they're reviewed here briefly for clarification. We have primary lateral sclerosis. It is categorized by its upper motor neuron pattern with little or no apparent lower motor neuron involvement. By some estimates, PLS is approximately 0.5% as prevalent as ALS. The most commonly reported differences in neuropathology between ALS and PLS lie in which regions have greater demyelination. PLS reportedly shows the greatest demyelination near the corpus callosum, whereas in ALS, demyelination occurs most in superior frontal gyrus. But PLS neuropathology does show changes in lower motor neurons, and these changes are of the same molecular pattern as is seen in typical ALS disease, including TDP-43 pathologies. Progressive muscular atrophy is characterized by its lower motor neuron pattern with little or no upper motor neuron involvement. ALS and PMA also share similar genetic mutations in familial cases. Despite the predominant involvement of lower motor neurons clinically, neuropathological studies have shown degeneration of the corticospinal tract, even in the absence of motor neurons, symptoms, or signs. Importantly, as is seen in ALS, inclusions positive for ubiquitin TDP-43 and FUS are frequently present. Therefore, the strongest evidence points to PMA being part of the disease spectrum and not a different disease. Overlap FTD. The clinical overlap of ALS and FTD is mirrored at the neuropathological level. Nearly all sporadic ALS and most familial ALS, except SOD1 and FUS, show TDP-43 proteinopathy, whereas only about 50% of frontotemporal lobar dementia is a TDP-43 proteinopathy. Most of the remaining cases of FTLD are considered as tauopathies, and a small percentage are FUS proteinopathies. The discovery of repeat expanded C9orf72 revealed a common genetic link between ALS and FTD and highlighted the fact that both ALS and FTD are phenotypes of disease as well as diseases. Spinal muscular atrophy, like PMA, is also characterized by degeneration of lower motor neurons and results from homozygous mutations in the SMN1 gene. SMA affects infants, juveniles, and young adults and is the leading genetic cause of infant death. Affected individuals exhibit muscle weakness and atrophy of muscle fibers. There is also extensive motor neuron loss, gliosis, neuronophagia, and chromatolysis in the anterior horn. Reduction in the number of BET cells in the motor cortex has also been observed. TDP-43 proteinopathy does not appear to contribute to SMA biology, at least in mouse models. Hereditary spastic paraparesis is a group of genetically heritable diseases that present with late onset, slowly progressive spasticity of the lower limbs. The neuropathological findings are almost entirely limited to the pyramidal tracts of the spinal cord and most significantly affect the longest ascending and descending axons. This axonal degeneration is particularly noticeable in the lumbar region of the spine. Some degeneration of the anterior corticospinal and spinocerebral tracts is also observed, as well as occasional loss of the cells in the anterior horn. Only one mutation of HSP has been studied neuropathologically and did show evidence of TDP-43 proteinopathy. Researchers are looking for aspects of lifestyle that can interact with genes to cause or contribute to ALS. Identifying and confirming that environmental factors are linked to ALS is extremely challenging, as the general population are all exposed to similar factors, and it is difficult to ascertain why one individual is susceptible to ALS, whereas another individual is not. BMAA, one of the first clues that ALS might involve environmental factor, was obtained on the island of Guam in the Pacific, where an unusually high proportion of people over the past century have developed symptoms similar to ALS as they age. Many theories have been explored and published over the years implicating environmental exposures or genetic susceptibility. One such toxin, BMAA, is suspected of causing amyotrophic lateral sclerosis and Parkinsonism dementia complex, a disease suffered by the Chamorro people of Guam. BMAA is a non-protein amino acid produced by cyanobacteria occurring in traditional dietary items consumed by the Chamorros. Feeding the bacterial neurotoxin BMAA to a variety of models systems leads to a protein aggregates and damages motor neurons, suggesting the toxin may contribute to disease found in humans exposed to the toxin. Investigations are ongoing to determine the role BMAA in the disease process in ALS. For smoking, a recent study established a link between smoking and developing ALS using rigorous evidence-based methods. Since smoking has no redeeming features, avoidance of smoking may reduce the occurrence of ALS in the future. Toxins. Many investigations of heavy metal exposure, particularly lead, mercury, and manganese, have been looked at as risk factors for ALS. A positive association between past exposure to heavy metals and risk of ALS has not consistently appeared across studies. These studies include occupational exposure that epidemiologists often use as a surrogate to assess potentially toxic exposures. Many of the studies relied on self-reporting by questionnaires. Self-reports can allow a recall bias. I have this disease, what did I do to produce it? That has the potential to artificially inflate the data. Some support for an association between ALS and exposure to organic solvents appears in epidemiologic studies, yet these have found only weak association and a mix of positive and negative results. As well, no consistent associations with ALS have emerged from investigation into the potential role of radiation and electromagnetic fields. In warfare, US veterans are another group of people who appear to develop ALS more often than the general population. Studies found that the relative risk of dying from ALS for veterans was approximately 2 times that seen for the men and women who did not serve. The increased risk was apparent for veterans of all wars, both deployed and undeployed, including World War II, the Korean War, the war in Vietnam, and the Gulf War. When it comes to exercise or pesticides, one idea that researchers offer is that soldiers on active duty are engaged in strenuous physical labor, or they are exposed to toxins which could play a role in ALS. Both possibilities of toxin exposure or the influence of intense exertion are supported when researchers consider the finding that Italian soccer players appear to have increased incidences of ALS. It remains unclear whether exercise is indeed a risk factor and what types of exercise may be of concern. Indeed, some studies seem to suggest that a degree of exertion may be beneficial in ALS. Other ideas are that pesticides or some other chemical encountered or unmaintained playing field might be involved. Viruses. A recurring idea about environmental influences on ALS is that a virus is responsible for the condition. Poliovirus, for instance, infects motor neurons and can at times lead to a weakening in condition called the post-polio syndrome, years after the initial infection. Scientists have considered viral infection for many other disorders of the nervous system, from schizophrenia to multiple sclerosis, as well as for ALS. A concrete link to viral infection has never been documented in any of these diseases. More recently, investigators published on the potential association of retroviruses with ALS, and this is still being further explored. ALS is a devastating disease for both people living with ALS, caregivers, and loved ones. Every person affected is different, and ALS presents in various ways. It can cause loss of motor neuron function in muscles from their legs, their arms, mouth, jaw, among other muscles that eventually leads to total paralysis. Ultimately, ALS affects their diaphragm, leaving people unable to breathe on their own. Assistive technology can include a variety of tools and devices that improve quality of life, for such as communication devices that allows people with ALS to communicate with ease, despite all their limitations. Examples of communication devices are speech-generating devices, electronic devices that synthesize speech, eye gaze control systems, devices that use eye movement to select letters and words on a computer to synthesize speech, writing tablets, devices used to communicate when people still have the ability to write, a brain-computer interface, a system that allows a person to control a computer or other electronic devices using only his or her brain waves with no movement required. They can be used for communication, computer access, or to control devices such as a wheelchair and prosthetic arms, among other applications. Text-to-speech computer software programs that consist of an on-screen keyboard and mouse that allows use without needing a person's hand. Text-to-speech apps are apps for use on smartphones or tablets that convert text to speech. Voice banking systems, devices that allow people with ALS to store the sound of their voice and recorded words and phrases before they lose their ability to speak. Communication devices are categorized into 2 segments, dedicated and undedicated. A dedicated device is strictly a communication device in that the only function it performs is speech generation. Undedicated devices, which are computer-based, not only speak, but also feature all the functions of a regular computer, such as word processing, email, internet access, among other applications. ALS treatments. There are currently 2 treatments approved by the US Food and Drug Administration for treatment of ALS, Riluzole and Edaveron. Riluzole was approved by the FDA in 1995 and has also been approved for marketing in many other countries such as Canada, Australia, and across Europe. Riluzole is thought to interfere with the activity of glutamate, one of the chemical messengers that transmit signals between the nerve cells. The medication is taken by mouth and may cause adverse effects such as dizziness, gastrointestinal symptoms, and liver function changes. Edaveron was approved by the FDA in 2017 and by Health Canada in 2018. The treatment is currently only available in the US, Canada, Japan, and South Korea, but is also under consideration by the European Medicines Agency. Edaveron is thought to counteract the excessive oxidative stress seen in ALS. When taken over a 6-month period, it has been shown to slow the rate of decline in ALS patients compared with a placebo. It is administered by infusion into a vein, typically for 10 to 14 days in a row, once a month. Side effects may include walking problems, bruising, and headaches, and serious allergic reactions to the sodium bisulfite in the formulation can also occur. It is to note that these 2 do not completely get rid of ALS, they slowly slow it down. There are also several therapies that people with ALS can go through. There are breathing cares, so patients will eventually have more difficulty breathing as their muscles weaken. Doctors might test their breathing regularly and provide them with devices to assist breathing at night. They can choose mechanical ventilation to help you breathe. Doctors insert a tube in a surgically created hole at the front of the neck leading to your windpipe, a tracheostomy that connects to a respirator. Physical therapy. A physical therapist can address pain, walking mobility, bracing, and equipment needs that help stay independent. Practicing low-impact exercises can help maintain cardiovascular fitness, muscle strength, and range of motion for as long as possible. Regular exercise can also help improve sense of well-being. Appropriate stretching can help prevent pain and help muscles function at their best. A physical therapist can also help adjust to a brace, a walker, or a wheelchair, and might suggest devices such as ramps that make it easier to get around. Occupational therapy. An occupational therapist can help find ways to remain independent despite hand and arm weakness. Adaptive equipment can help perform activities such as dressing, grooming, eating, and bathing. An occupational therapist can also help modify home to allow accessibility if there is trouble walking safely. Speech therapy. A speech therapist can teach adaptive techniques to make your speech more understandable. Speech therapists can also help explore other methods of communication, such as an alphabet board or pen and paper. Patients should ask the therapist about the possibility of borrowing or renting devices such as tablet, computers with text-to-speech applications, or computer-based equipment with synthesized speech that can help communicate. Nutritional support. The team will work with the patient and their family members to ensure they're eating foods that are easier to swallow and meet nutritional needs. They might eventually need a feeding tube. Psychological and social support. The team might include a social worker to help with financial issues, insurance, and getting equipment and paying for devices they need. Psychologists, social workers, and others may provide emotional support for them and their families. Statement 2. Cytoplasmic aggregated proteins, non-enzymatic adaptive proteins, are a common neuropathological feature of neurodegenerative diseases. Cytoplasmic mislocalization and aggregation of TDP-43 is found in the majority of patients with ALS and in approximately 50% of patients dying of FTLD. Prudencio Humphrey Pickles and their colleagues investigated the relationship of TDP-43 pathology with the loss of stamin 2, which is an essential protein for axonal growth and maintenance. Comparing genetic, cellular, and neuropathological data from patients with TDP-43 proteinopathies and FTLD with data from patients with non-TDP related neurodegenerations, they demonstrate a direct relationship between TDP-43 pathology and stamin 2 reduction. Loss of the normal transcription suppressor function of TDP-43 allowed transcription of an early termination cryptic axon, resulting in truncated, non-functional messenger RNA. The author suggests that measurement of truncated stamin 2 mRNA could be a biomarker for discerning TDP proteinopathies from other pathologies. Taking this into fact, TDP-43 is also a transcription suppressor. In the absence of TDP-43, for example, when TDP-43 is cleared from the nucleus as it mislocalizes to the cytoplasm, cryptic exons are exposed and transcribed, creating aberrant mRNAs that lead to neurotoxicity in ALS models. Recently, this TDP43 suppressor function was specifically implicated in suppressing a cryptic early polyadenylation site in the pre-mRNA for the protein stamin 2. Loss of TDP43 allows incorporation of a premature poly A tail, resulting in a truncated and non-functional form of stamin 2. Stamin 2 is a highly conserved cytosolic protein essential for axonal outgrowth and maintenance. Stamin 2 knockout mice develop late onset, predominantly motor axonopathy, paralleling that seen in human ALS. Amyotrophic lateral sclerosis is a fatal neurodegenerative disease that imposes a great burden on the patient's family, and society. However, we lack information about the total disease burden at a national level, especially regarding costs before and after diagnosis and the consequences for spouses. They aimed to estimate the factual, direct, and indirect costs of ALS in a national sample. Using records from the Danish National Patient Registry from 1998 to 2009, 2,394 patients with ALS were identified and subsequently compared with 9,575 randomly chosen control subjects matched for age, gender, and geographic area and civil status. Direct costs, including frequencies of primary and sector contacts and procedures, and medication from primary and secondary sectors were obtained from the Danish Ministry of Health, the Danish Medicines Agency, and the National Health Security. Indirect costs included labor supply and social transfer payments and were based on income data derived from coherent social statistics. Patients with a diagnosis of ALS had poor survival. The average 5-year survival rate was 0.278, compared with 0.865 among controls. Patients with ALS had significantly higher rates of health-related contact and medication use and higher socioeconomic costs. They also had very low employment rates, while those in employment had a lower income level than control subjects. The annual mean excess health-related cost was 18,918 euros for each ALS patient. However, spouses showed no excess health usage. In fact, their employment and income rates were higher, and the net cost was reduced by 3,420 euros. They concluded that ALS has a serious mortality, health, and socioeconomic consequences for patients. However, the consequences for spouses are complex. They tend to compensate for the social consequences to patients by increasing their net income after ALS diagnosis. In clinical trials, we have observational clinical trials. This is a type of trial in which enrolled participants are observed. The outcomes are measured, such as measures of strength or function may be part of the observation. No treatments are given, often used to learn about trends of symptoms or the course of a disease, or to find a disease biomarker. These trials include natural history studies. Interventional clinical trials is a type of trial in which exposure to the potential therapy being tested, such as a drug, is assigned. It is used to determine the effectiveness of a treatment or an intervention. Due to requirements for producing reliable data, patients may have to be of a certain age, gender, stage of disease, or even race to participate. People living with ALS participating in clinical trials are required to fulfill certain criteria of the disease or disease severity, and might be ineligible if they show certain other characteristics. Clinical trials can only test relatively few patients and must make predictions from a few people that will most likely hold for the patient population as a whole. Statistics provide the means to judge if a change induced by drug treatment is likely to be a real reproducible change, and for doctors to understand whether the observations in a restricted set of patients is likely to hold true for the entire population of patients with that disease. In the pilot trial, it is a small study that may include testing of an approved drug for another indication that is being tested in ALS patients to determine safety and tolerability, or a study to identify relevant biomarkers. Phase 1 testing looks at the safety of a candidate treatment, often in 20 or fewer people. Participants are examined for any adverse reactions or side effects. If any appear that are judged to be too dangerous, testing is halted and the drug will not advance any further in the clinical trials process. Phase 2 testing attempts to determine the optimal dose, route, and timing of doses of the candidate treatment. Information about a drug's ability to help in the disease may be obtained in the course of phase 2 testing, but such findings are not able to reliably predict the candidate's effect. Usually for ALS, less than 100 patients are involved. Phase 3, the therapeutic effect of the candidate is specifically sought in phase 3 testing. This is the stage of testing that enrolls enough patients to allow statistical judgment that a treatment is effective. Phase 3 trials for ALS usually require hundreds of participants. Large numbers are required due to the diversity of the ALS population. No person with ALS is the same, and notably, disease progression from one individual to another is highly variable. So one clinical trial is the identification of genes causing familial ALS or increasing risk for sporadic ALS and ALS with frontotemporal dementia and understanding disease mechanism. It is being conducted by Tipu Siddiq, MD, at Northwestern University. The investigators' long-term goals are to improve diagnosis and develop effective treatments that arrest or ameliorate symptoms of ALS and possibly delay or prevent disease onset in individuals at risk for developing familial ALS. In order to do this, one must understand how disease develops at a molecular level. Identification of genes that increase risk for developing all types of ALS will reveal the pathways of molecular events that are involved in ALS. The investigators are collecting blood samples, family and medical histories of patients with all types of ALS, familial and sporadic, with and without frontotemporal dementia, and primary lateral sclerosis and particular family members. Samples are coded to maintain confidentiality, and travel is not necessary. Stem cell research. Embryonic stem cells have the ability to differentiate into all germ layers and serve as a source for both cellular replacement in ALS and for disease modeling when differentiated into motor neurons. Early studies examining intraspinal transplantation of embryonic stem cells derived from motor neurons into G93A, SOD1, glycine 93 changed to alanine, rats demonstrated transient functional improvements. However, there was no apparent axonal projections to the periphery, no effect on neuromuscular junction formation, no long-term effects on the lifespan of the rats, and limited graft survival. Given that mesenchymal stem cells do not naturally differentiate into neural lineages, the use of mesenchymal stem cells as a vehicle to deliver neuroprotective factors to the central nervous system. Intracerebroventricular injection of G93A SOD1 mice with mesenchymal stem cells expressing glucagon-like peptide 1, a peptide with antioxidant properties, confers significant effects on the disease course, including a 15-day delayed onset, a 13-day improvement in survival, and attenuated neuroinflammation, astrocytosis, and microglial activation. Intramuscular injection of mesenchymal stem cells expressing increased levels of glial-derived neurotrophic factor, improved motor neuron health, and extended survival by 28 days in G93A SOD1 rats. Neural progenitor cells are emerging as a promising cellular therapy for clinical translation in ALS. In ALS models, motor neuron degeneration triggers endogenous NPC niches in the CNS to proliferate, migrate, and promote neurogenesis in the spinal cord as a natural response to the disease. However, the limited number of endogenous NPCs is likely insufficient to combat the toxic, progressive degeneration associated with ALS. Thus, NPC cell lines with robust growth properties and neurogenic potential have been developed, and transplantation of NPCs has been extensively studied as an attempt to augment this natural defense mechanism. Potential mechanisms of stem cell efficacy in ALS. In addition to motor neurons, multiple cell types within the motor neuron microenvironment play a role in disease pathogenesis. Therefore, the delivery of stem cell-based therapies, in blue, have the potential to provide support through many different mechanisms. Within the spinal cord, stem cells that differentiate into neurons, green, can synapse with existing motor neurons to reestablish or maintain neurocircuitry, as we can see in cell A, as well as provide neurotrophic support, as we can see in cell B. Differentiation of stem cells into non-neuronal cell types, yellow, within the spinal cord microenvironment can also impact disease progression by providing neurotrophic support and attenuating oligodendrocyte dysfunction and mitigating toxicity, as you can see in C. In the periphery, stem cell transplantation into muscle can provide critical support to maintain functional neuromuscular junctions, as you can see in D. Finally, the mobilization of endogenous mesenchymal stem cells from the bone marrow into the circulation can also induce immunomodulatory effects that attenuate inflammatory responses within the spinal cord via the production of cytokines and other anti-inflammatory mediators, as you can see in E. CoreStem is a biotechnology company specializing in the research and development of personalized stem cell therapies for neurological and autoimmune diseases. Its lead product is Neuronata R, the world's first stem cell-based therapy for amyotrophic lateral sclerosis. Neuronata R is based on autologous bone marrow-derived mesenchymal stem cells. Treatment involves extracting bone marrow from the patient, isolating and culturing mesenchymal stem cells, mixing with cerebrospinal fluid collected from the patient, and administering the final product by intrathecal injection. The first Neuronata R injection typically takes place 4 weeks after the first bone marrow extraction, followed by a second injection 4 weeks after. Treatment with Neuronata R results in various effects that prevent motor neuron death and slow the progression of ALS. These include the release of immune modulating factors such as regulatory T lymphocytes and T helper 2 factors, a motor neuron protective effect through the expression of growth factors such as brain-derived neurotrophic factor, and an anti-inflammatory effect due to microglial cells switching from an M1 to an M2 phenotype. Data from clinical studies show that Neuronata R slows the progression of ALS as measured by a change in ALS Functional Rating Scale Revised, ALS-FRSR. A phase 1 open-label study demonstrated the safety and feasibility of 2 repeated intrathecal injections of autologous bone marrow derived from mesenchymal stem cells over 12 months in 7 patients with ALS. This was followed by a phase 2 parallel group randomized controlled trial of 2 repeated intrathecal injections, which demonstrated safety and effectiveness for slowing the progression of ALS for at least 6 months. On the basis of phase 1 and 2 data, Neuronata R was approved in South Korea in July 2014 and launched in February 2015. By March 2019, 371 patients had received Neuronata R in South Korea. CoreStem is now preparing to conduct an innovative randomized placebo-controlled multi-center phase 3 study to assess the safety and efficacy of Neuronata R in more than 94 patients with ALS. Patients will be randomized to receive 2 doses of study drug or placebo, and a cryopreservation procedure will be developed so that ALS patients do not have to undergo repeat procedures for bone marrow extraction. Thank you.
Thank you, Dr. Michael Bassil. Is it okay if I ask a few questions about your presentation? That was amazing, by the way. It was amazing, really good presentation. Is it okay with you?
Yes.
Okay. One of my first questions would be if and is ALS common among men and women?
Yes, so actually
equally?
It is slightly more common in men than women, but recent studies suggest that this difference is decreasing over time. And familial ALS is equally common in both men and women.
And do clusters of ALS probably exist?
So actually, it is unknown whether clusters of ALS actually exist. Although it is possible that cases of ALS are related, studying clusters does not help when trying to determine what factors cause or trigger ALS. Yeah.
And what are the problems when studying possible clusters of ALS?
So the cause of most cases of ALS is unknown. So we don't know what to look for in a person's past. We do not have a current or complete data on the number of people with ALS. And as a result, we cannot determine the normal or expected number of cases among a group of people in a specific area. So the expected number of cases of a disease is often found in an existing disease registry, such as with cancer. However, there are no registries or ongoing tracking efforts for ALS in the United States.
Okay, thanks so much for answering my questions. I want to thank you once again for participating today in this amazing project. And I want to thank everyone involved in the Congress. And thank you to all my colleagues that are here today with us. Have an amazing day. Thank you so much, Dr. Bassil.
Thank you for having me.
Thank you.
Have a good day.
Thank you. Have a good day.